Okay, the next speaker is Dr. Cynda Crawford. And Dr. Cynda Crawford is the Frederica Salzman Endowed Professorship Chair in Shelter Medicine at the University of Florida College of Veterinary Medicine. Her areas of expertise include canine and feline infectious diseases and evaluation of diagnostic tests and vaccines for infectious diseases. Career accomplishments include identification of the H3N8 canine influenza virus, development of canine influenza vaccines, development and validation of diagnostic assays for canine influenza virus, and epidemiological studies on canine influenza virus transmission in dog populations. Ongoing work includes evaluation on different intervention strategies for preventing or controlling transmission of canine respiratory viruses in animal shelter populations. And Dr. Crawford, thanks for joining us, and please proceed with your presentation.
Thank you very much. Good morning, good afternoon, and good evening to our international audience. I am going to present a barnyard story for you. I am a veterinarian, so this is kind of what I am very comfortable with, is talking about chickens, cows, and cats. And this presentation will focus on the impact of bird flu on these animals. So Dr. Webby's already introduced us in an earlier presentation about what is bird flu. And there are many influenza A viruses that infect avian species, but the one that we are most concerned about and focused on for this presentation is the H5N1 subtype of avian influenza virus. Now, wild birds, ducks, and geese, waterfowl in general, are the natural hosts for all influenza A viruses. And most of the time, these viruses are not pathogenic to their hosts. But other influenza viruses, such as H5N1, have acquired characteristics that make them highly pathogenic in natural hosts. So we call them high-path flu viruses. Now, H5N1 was first discovered in geese in 1996 in China, and I think the first human infection by this avian influenza virus was in a person in China the next year. H5N1 avian flu is an old virus, actually. It's been circulating in birds and poultry around the world now for the past 29 years. Of course, during this time, it has gone through several reassortment events and mutations to alter pathogenicity and host specificity. So the U.S. historically has not been much of a hotspot for H5N1 avian flu infection of birds, but in 2021, it was inundated for the first time in our history with a invasion of H5N1 clade 2.3.4.4b high-path avian flu from Europe to North America, spread through migratory birds. And here is a map of counties in all 50 states of the US where H5N1 bird flu has been detected in wild birds, ducks and geese, since 2022. So it's very pervasive throughout the US. Again, this is a first time event for this virus in this country. The H5N1 avian flu spilled over into poultry from the wild bird populations in 2022. And to date, poultry in all 50 states have been infected by H5N1. As this map shows, there are infected flocks in every state, with some states such as California, South Dakota, Minnesota, Ohio, and Pennsylvania overrepresented by infected poultry. There's estimated to be almost 175 million birds, poultry, and different types, chickens, turkeys, that have been infected. These birds resided in 1,700 commercial flocks, as well as backyard flocks where chickens, other poultry species are kept as pets and for eggs. And this has resulted in depopulation of most infected birds, more than 175 million. This is a virus that is devastating to poultry in that it causes a very severe pneumonia, and basically poultry will suffocate to death when they become infected by H5N1. And virtually all birds will become infected upon exposure. So it is considered most humane to depopulate flocks once infected birds are identified because of the severe disease. And after the spillover of H5N1 from wild birds, ducks, and geese to commercial and backyard poultry, there was about a month later a spillover to mammals. And this was associated with a reassortment event of the 2344B clade viruses to create genotypes, different genotypes within that clade capable of infecting mammals. These new genotypes have the capacity to bind not only to the alpha-2,3 sialic receptors on avian host cells, but also to alpha-2,6 sialic acid receptors found primarily on mammalian host cells. And many mammalian species have a mixture of alpha-2,3 and alpha-2,6 sialic acid receptors in their respiratory tract. So this map shows the different species of mammals that have been infected in the US, where the key over here shows the mammals referred to where this virus has been identified as a source of infection. More than 200 terrestrial and marine mammalian species so far have been infected by H5N1 bird flu. And this includes rodents, raccoons, foxes, other carnivores, coyotes, and then as Dr. Webby pointed out in his presentation, this virus has infected mammalian species that have not been thought to be a host for influenza in the past, and that's the seals and sea lions. This H5N1 virus is called massive die-offs of seals and sea lions in both North America and South America. What this map shows is, in addition to the clustering of infected mammals of different species from coast to coast, is an overlay of the migratory bird flyways with the Pacific Flyway and the Central Flyway, Mississippi Flyway, and Atlantic Flyway. So most of the cases of infected mammals seem to be clustered in the Pacific, Central, and Mississippi flyways, heavily traveled by migratory birds twice a year. And this could be maybe the source of spillover to mammals during these migratory routes. Then something else unusual happened in March 2024. Yet another species of mammal was found to be infected by H5N1 avian flu virus. So we've already identified marine mammals as an unusual host for bird flu. And now it was identified in a herd of dairy cattle in the US in March 2024. The cattle were infected with yet another reassortment, the clade 2344B H5N1 virus, to create a genotype called B3.13. This genotype was found to also be circulating in wild birds located in geographical areas where the infected dairy cattle were identified. And the first detections were in dairy farms in Texas, Kansas, and Arizona. We do not really know how this avian influenza virus first infected cattle and why dairy cattle and not beef cattle, but it's speculation that the cattle ingested feed that was contaminated by feces from infected wild birds, since these barns are open to the environment and there are lots and lots of birds that hang out and roost in dairy barns. In early 2025, there was the emergence of yet another unique genotype called D1.1 in dairy cattle in farms in Nevada and Arizona. Again, this genotype, D1, was also found in wild birds and in poultry in the same areas. What's different about the D1 genotype from the B3 genotype is that the end gene sequence of D1 is totally different from the end genes in any of the other genotypes in the H5N1 clade 2344B. So this is a very unique reassortment producing a unique neuraminidase gene sequence. So H5N1 is a localized viral infection in dairy cows. It is not a respiratory pathogen. It has not been identified in the respiratory tract tissues or any other tissues in the cow. It seems to be very localized to the mammary gland of all places. It is not known why this virus localizes to the udder or mammary gland, but the result is mastitis or severe inflammation of the mammary gland. And you can see the red area of this udder that is severely inflamed compared to this more normal part of the udder. The mastitis causes a very sudden or abrupt drop in milk production. The milk is off-colored. It's no longer the nice white color. It becomes a very creamy yellow. And testing of milk from these infected mammary glands has shown very high viral loads of infectious virus in the milk. Now, this is not a fatal infection in dairy cows compared to wild birds and poultry and marine mammals and other mammals. It's a transient infection. Eventually, the cow's mastitis resolves, milk production returns to normal, and there's no lasting effects from the avian influenza virus infection, and the milk clears up. Virus is spread by movement of infected cows between dairy farms, also by movement of contaminated milking equipment, and by direct contact with milk spillage from infected cows. So, going to Dr. Moroska's talk, there's been recent discovery of infectious respiratory particles. I guess that's the terminology to use now, not aerosolized virus. But putting milking machines on the udders of hundreds of cows being milked all at the same time in the same location twice a day generates a lot of infectious viral particles in the air during this process. And this could be how this virus is transmitted between cattle and between cattle and people, the dairy workers in the milking parlor at the time. Studies have also shown that runoff water used in the barns to clean the milking parlors and the equipment contains infectious virus accumulating from milk spillage from cows with infected mammary glands. So there are several transmission routes for H5N1 bird flu virus between farms, between cows that have not been recognized before. So this virus has significantly impacted the dairy industry in the US. This is a map that shows the 17 states where infected dairy cattle have been identified. In addition, the transmission routes that I just discussed, the contact with milk, contaminated machines, airborne particles generated during milking, and runoff water containing infectious virus, has managed to sustain H5N1 circulation within dairy cows, and it has spilled over to infect dairy workers and dairy veterinarians. As Dr. Webby told us earlier, the H5N1 genotype B3 infections acquired by dairy workers and dairy veterinarians are mild. Some of the infections have been subclinical. Usually it's a clinical disease is manifested as conjunctivitis, and it's very transient. And so far, there's been no evidence that dairy workers and dairy veterinarians can transmit their infection to other people or back to cows. So this brings me to cats. Can cats catch bird flu? We have known now for 20 plus years that cats are sensitive or susceptible to infection by influenza viruses, specifically H5N1 avian influenza. And hundreds, if not thousands, of infected cats were identified during H5N1 circulation in Asia and Europe from 2004 to 2006. And these infected cats were geolocated in areas where there was known infections in wild bird and poultry. It was shown that most of the cats acquired H5N1 influenza infection through ingestion of infected bird and poultry carcasses. It's very common in Asia and certain European countries for cats, outdoor cats, free roaming cats, barn cats, to be intentionally fed carcasses from birds and poultry. And so this has been shown to be a common route of acquisition of infection by cats. In contrast to dairy cows, infection of cats causes a very severe neurological disease that is fatal. There was a lull, and actually no reports of H5N1 infected cats from 2007 to 2021, but with resurgence and reemergence of the H5N1 influenza virus in Europe in 2021, with spread to other countries in Europe and then into the US, we are seeing more cases of cats infected by the new H5N1 clade 2.3.4.4b virus. So the cat story begins with the cow story, because in March 2024, on the same dairy farm in Texas where the first cattle were identified as infected by H5N1, there was a cat die-off. This particular farm, and this is actually a picture of the cats that lived on this dairy farm, the barn cats. Here they are in the milking parlor area of this dairy farm, and they are drinking milk offered to them. And this is raw milk. This is not pasteurized milk. This is fresh milk collected from the dairy cattle. And they were unintentionally fed milk from some of the infected cows. The cows weren't actually known to be infected by H5N1 at that time. More than 50% of the cats died within 2 days. Postmortem examinations on some of the dead cats revealed a systemic infection with a severe encephalitis or infection of the brain, pneumonia, myocarditis, and H5N1 flu virus was recovered from the brain, lungs, and heart of cats that died. And remember, the infection in cows was very unnoticeable almost, except for the fact that they had a very sudden drop in milk production, in contrast to cats with a severe systemic high fatality infection. The cows only had a transient mastitis, localized infection. Whole genome sequencing of virus recovered from cow milk on this farm, and virus recovered from tissues of the cats that died on this farm revealed a match in the sequences belonging to clade 2.3.4.4b. So this is the first report of H5N1 avian flu virus transmission via ingestion of raw milk. There's lots of reports and documentations of transmission via ingestion of virus-contaminated meats and carcasses, but ingestion of raw milk is a new event or route for mammal-to-mammal transmission. So H5N1 avian flu has not only spilled over to mammals, spilled over to dairy cattle, it has spilled over into cats in the US. This is a map of the different mammalian species identified with H5N1 infection. The cats are the little black boxes in this map. There are, let's see, clusters of infected cats along California, Oregon, and Washington. That's a lot of dairy cows, farms located in this section of the country, as well as lots of infected poultry and wild birds. Many of the cases also in the midsection of the country. Again, this is dairy farm country. Lots of infected poultry and infected wild birds. And there is one cluster of 6 infected cats on the East Coast, but so far no identification of infected cats other than this cluster on the East Coast. So 23 states, 137 cats and counting, and calculated mortality rate of 50 to 70% so far. So what cats are at risk for bird flu? Obviously, feeding cats raw milk, in this case directly from the teat, is a big risk factor. Cats with outdoor access in locations where H5N1 is known to be circulating in birds and other mammalian species, rodents, raccoons, other wildlife reservoirs. Obviously, cats living on dairy farms, poultry farms, or premises with backyard flocks of chickens are at risk. And then it is thought that cats could be infected by exposure to contaminated farm workers and their clothing if farm workers from infected dairy or poultry premises wear their clothing home and handle their pet cats. Lots of poultry farms have barn cats, and chickens and cats get along very well. Other cats at risk for contracting bird flu are those with outdoor access and access to infected mice and dead birds, so predation. And then this is a new route of virus transmission, and that's through consumption of unpasteurized dairy products, feeding raw poultry-based diets to cats, or feeding undercooked poultry. And there are many documentations of indoor-only cats with no outdoor access actually acquiring H5N1 flu through eating raw milk and raw poultry diets acquired from commercial vendors. It just so happens that these manufacturers of these raw diets, which are hugely popular to cat owners for feeding their pets, these local manufacturers are located in geographic hotspots where there are infected cows and poultry. So they are sourcing those infected animals for the diets that they're then selling to cat owners. And here are some of the symptoms that have been seen in infected cats so far. Again, a severe neurological disease, meningoencephalitis. It causes clinical signs that very much mimic rabies. So a cat infected by H5N1 avian influenza virus will look very much like a cat infected by rabies virus. And this certainly creates a diagnostic dilemma and conundrum for veterinarians. These cats can also have a concurrent respiratory infection, and the median time from illness onset to death is only 2 days, a very quick hit. And then I'll wrap up by asking the question, what is the evidence that cows and cats can spread H5N1 influenza infection? Well, cows shed virus in their milk, again, a very localized infection, transient, but high viral loads. Sustained cow-to-cow transmission by contact with contaminated milk and equipment, infectious respiratory particles created during milking, and contaminated runoff water all maintains the virus circulation in a dairy cow herd. And there are documented cases of cow to human transmission. but no subsequent human to human transmission. So cats shed virus in urine and respiratory secretions. It's a systemic infection. To date, there's no evidence of cat-to-cat transmission or cat-to-human transmission. So it may be, in contrast to poultry and cows, that cats are a dead-end mammalian host. Don't know. So here's the new H5N1 avian flu ecology as of 2025. We have the wild waterfowl reservoir for the highly pathogenic influenza A viruses. This is the origin for infection of migratory birds as well as poultry flocks. The H5N1 clade 2, 3, 4, and 4B viruses can infect pinnipeds, marine mammals, cause massive die-offs, more than 200 species of mammals, in addition, are infected by this new clade of H5N1. Now we've added dairy cows. First time that a ruminant has been shown to be infected by an avian influenza virus. We have cats also involved in the ecology of H5N1 flu virus. And so far, humans have acquired infection from infected poultry and infected cattle, but no human to human transmission. So this brings up public health concerns. Can cats and cows serve as incubators for evolution of a more pathogenic virus genotype that can spill over and easily infect people and cause severe disease? In other words, are cows and cats the new mixing vessel for H5N1 avian influenza virus, as opposed to us always talking about pigs or swine being mixing vessels for creation of a new novel influenza genotypes? So there's certainly some characteristics of the new H5N1 genotypes that cause concern for cows and cats being mixing vessels or incubators for generation of new pathogenic genotypes. Frequent reassortments are always occurring. The reassorted viruses can bind to both avian and mammalian receptors. And cows and cats have a lot of close contact with people. So we need to have ongoing surveillance with H5N1 testing and sequencing of suspect cows and cats to identify new genotypes with possibly novel transmission routes. Of course, that brings up questions about who performs this surveillance. Are there individuals? Is it the farmers? Or is it veterinarians? Is it the government or regulators? Who bears the cost of surveillance? Is it the public? Is it private ventures, or is it the government? And then vaccination. Certainly, as Dr. Webby said, there are already stocks of H5N1 vaccines for people and for poultry. Yes, there is an H5N1 vaccine ready for dairy cows, and there's an H5N1 vaccine that can be quickly developed for cats using an mRNA platform. So this will be something that is going to be discussed quite intensely over the near future as to whether this is a plausible or feasible or practical approach for control of H5N1 influenza infection in mammalian species that have close contact with people and could possibly serve as incubators. Do we need to use vaccines to protect these incubator species? So this is a One Health problem. Certainly veterinarians are on the front lines for identifying infections in cows and cats and other species of veterinary interest. It's our job to communicate with the public health sector, educate farmers and pet owners. Some things that can be done with dairy cows is to increase testing. Should it be mandatory? Should it be regulated by the government? There is huge resistance of dairy farmers to allow testing of their cattle or their farm workers. Should we regulate movement of cows and equipment between farms? Right now, there is no real regulation, so there could be transfer of infected cows and supplies and equipment. And then, of course, farm workers should shower, change clothes, and wash their hands before leaving farms with infected cattle and before going home and having contact with pets and family. Cats, be nice if they were just kept indoors or at least in an enclosed outdoor facility, but that is not the wish of all cat owners. Many cat owners in the US as well as Europe and Asia feel that cats live their best lives if they are indoor, outdoor, and have a choice. But we really have to think about those that do have outdoor access, reducing potential contact with birds, rodents, poultry, and dairy cattle. And then the least we can do is stop feeding raw milk and raw poultry diets to cats, whether they're outdoors or whether they're strictly indoors. This is something that should not be done. Has been shown over and over again to be a source of H5N1 infection to cats, as well as other types of pathogens found in these raw diets. And with that, I have provided a list of selected references containing the material that I presented here. And I thank you for your attention.
Well, thank you, Dr. Crawford. That was quite amazing in the depth and breadth of the presentation. Just a quick question. It seems like you said that there's a wild canids, I guess that's foxes and coyotes or something that turn up with it. But there hasn't been any dogs turning up with it on the farms yet?
No,
That's
despite
interesting.
some testing, there's not been any reported cases of H5 clade 2344B in dogs in the US. Now, dogs have been infected by H5N1 avian influenza during the 2004-2006 circulation of H5N1 in Europe and Asia. And those dogs acquired infection just like cats in Eurasia through ingestion of carcasses of infected birds and poultry. So
Wow.
yes, they can be infected.
But it wasn't on your map there of all the different.
No, that has not been identified in the domestic dog yet
Wow.
in the US.
Thank you very much. Fascinating talk.